All right, welcome. Today, we are taking a really deep dive into the powerhouses of our cells, the incredible mitochondria. So here's our game plan. We're going to start with why everyone calls them the cell's powerhouse. Then we'll get into their anatomy, see what they're actually made of. After that, we'll watch how they forge cellular energy, and then uncover the secrets of their very own private genome. We'll also look at what happens when these powerhouses fail, and finally, we'll solve the puzzle of how they get all their workers. Let's get started. So first things first, let's establish what mitochondria are and, why they are so unbelievably critical for pretty much all eukaryotic life. Everything from tiny single-celled yeast to complex creatures like you and me.
You probably heard this analogy a million times, and for a really good reason. Their main job is to generate almost all of our metabolic energy through a process called oxidative phosphorylation. They take the fuel from things like carbs and fats and turn it into adenosine triphosphate, or ATP, which is the universal energy currency that powers, well, just about everything a cell does.
But, and this is a big but, there's so much more than just simple power plants. They're this fascinating hybrid system, almost like a living relic of an ancient bacterial past. You can see this in their semi-autonomous nature. They have their own circular DNA. This leads to this really unique dual origin system for their proteins. A few are made right there on site using their own genetic blueprint, but the vast majority, we're talking over 95%, are actually coded for by the cell's nucleus, built out in the cytoplasm, and then carefully imported inside.
So how in the world is this organelle built to perform such a critical role? To really get its function, we've got to explore its intricate architecture. And this double membrane system, it's the key to absolutely everything that mitochondria does.
And this just shows it beautifully. You can see the 4 key compartments. First, you've got a smooth outer membrane. Then there's the intermembrane space. After that, you have this highly folded inner membrane. And those folds, those are called Cristae. And their whole purpose is to dramatically increase the surface area. And right at the very center is the matrix, which is this dense, fluid-filled space that holds all the enzymes and, of course, the mitochondrial DNA.
Now, what's really fascinating here is the massive difference between the two membranes. The outer membrane is basically a sieve. It's super permeable because of these proteins called porins, which let small molecules just pass right through. But the inner membrane, it's the complete opposite. It's incredibly selective, totally impermeable to ions, and just packed with protein, over 70% protein. This selective barrier is absolutely crucial. It's what lets the mitochondrion maintain a steep electrochemical gradient, which is the driving force behind making ATP. This inner membrane isn't just a wall, it's the stage where the grand finale of energy production happens.
So, how does this amazing structure actually produce the energy our cells need to survive? Let's follow the entire process, a journey that actually kicks off out in the cell cytoplasm and ends deep inside the mitochondrion.
Think of it like a 6-step metabolic assembly line. It all starts with glucose breaking down into pyruvate out in the cytosol. That pyruvate then gets shipped into the mitochondrial matrix, gets converted to something called acetyl-CoA, and then it's fed into the citric acid cycle. The output from that cycle then fuels the electron transport chain, which creates the power for the very last step, making tons of ATP.
Right here, inside the matrix, the citric acid cycle grabs that acetyl-CoA and just systematically tears it apart. Now, the main goal here isn't really to make ATP directly. No, it's to load up high-energy electron carriers. These are molecules like NADH and FADH2, and they've basically captured all the high-energy electrons from breaking down acetyl-CoA. These carriers are the essential fuel for the next and most productive stage of the whole process.
So here's the absolute crucial point. The protein complexes in the electron transport chain use the energy from those electrons to pump protons out of the matrix. This builds up a massive electrochemical gradient. Seriously, think of it like water building up behind a gigantic dam. It's a huge store of potential energy. The only way for those protons to flow back down the gradient is through this incredible molecular machine called ATP synthase. And that powerful flow of protons is what physically spins the machine, allowing it to just crank out massive amounts of ATP. Okay, let's switch gears and talk about one of the most unique features of mitochondria. The fact that they have their very own separate and distinct genetic system, a direct link to their evolutionary past. And here it is, the human mitochondrial genome. It's a small circular DNA molecule, just like a bacterium's. This is some of the strongest evidence we have for the endosymbiotic theory, the idea that mitochondria were once free-living bacteria that got swallowed by an ancient cell.
In us humans, this tiny genome is packed with genes for 13 essential proteins for the electron transport chain, plus all the RNA machinery needed to build them, 2 ribosomal RNAs and 22 transfer RNAs. And get this, while the human version is tiny at only about 16,000 base pairs, those are the fundamental units of DNA. Its size just varies enormously across different species. Yeast's mtDNA is about 5 times larger, and some plants have mitochondrial genomes more than 10 times larger than that. It just shows you what a fascinating and diverse evolutionary path they've all taken.
Now, because mitochondria have their own DNA, they are susceptible to mutations, and these mutations can have absolutely devastating consequences for our health, especially in tissues that are really energy hungry, like the brain, the heart, and our muscles.
A really key feature of these mutations is that they are transmitted maternally. See, since the sperm contributes basically no mitochondria to the fertilized egg, your mitochondrial DNA comes entirely from your mother. This is super important for diagnosing diseases like Leber's hereditary optic neuropathy. But beyond specific inherited diseases, the slow, steady buildup of mutations in our mitochondrial DNA over a lifetime is now thought to be a major driver of the aging process itself. Why? Because these mutations can cause a decline in energy production, which contributes to the functional decline we see in aging tissues.
This comparison is just wow, it's so striking. The mouse on the left is a normal, healthy mouse. The one on the right has a single defect in a nuclear gene that codes for its mitochondrial DNA polymerase. This faulty enzyme makes a lot of mistakes, causing mtDNA mutations to pile up really fast. And the result? The mouse shows clear signs of premature aging, graying fur, hair loss, reduced muscle mass. It's a powerful and direct illustration of the link between mitochondrial health and the aging process.
So this brings us to a really critical question. If the mitochondrial genome is so tiny, it only codes for 13 proteins, where do the other 1,000 or so proteins it needs to function actually come from?
Just pause and think about that for a second. Over a huge amount of evolutionary time, most of the original mitochondrial genes actually got transferred to the cell's nucleus. The solution to this evolutionary puzzle is a super sophisticated and absolutely essential import system that bridges the gap between the nucleus and the mitochondrion.
And here's the roadmap for that whole import process. Proteins that are destined for the mitochondria get built on free ribosomes out in the cytosol. They carry a special tag at their front end called a pre-sequence. It's a sequence of about 20 to 35 amino acids that basically acts as a molecular shipping label, directing them straight to the receptor complexes on the outer mitochondrial membrane.
This whole journey is this carefully choreographed series of handoffs. First, that pre-sequence is recognized by a receptor on the outer membrane called TOM20. Then, the protein is threaded in a completely unfolded state through a channel called TOM40. Once it's in the inner membrane space, it gets passed to a second complex on the inner membrane known as TIM23. The electrical potential across this inner membrane acts like a magnet, pulling that positively charged pre-sequence right into the matrix. Finally, once it's inside, that targeting sequence is snipped off, and the protein folds into its final active shape.
So you might be wondering, how does the protein get pulled all the way through without just slipping back out? Well, inside the matrix, a chaperone protein called mitochondrial HSP70 is waiting, and it acts like a molecular ratchet. As the protein chain starts to emerge, HSP70 grabs onto it and, using the energy from ATP, it yanks it forward, preventing it from backsliding. This makes sure that the import process is strictly a one-way street. So from generating the energy that powers our every single move, to carrying its own unique genetic legacy, and to its profound role in disease and aging, the mitochondrion is just so much more than a simple powerhouse. It's a dynamic, complex player in the life of the cell. And its many secrets continue to define the frontiers of modern cell biology, offering these really tantalizing possibilities for understanding and maybe even one day influencing the processes of aging and disease.